The broad interest of the lab is in insect flight. And so one of the things that we look at is things like chases. So for instance, you can see here a house fly. This is not a moth, this is a fly. But you can see that it's taking off, and there's another insect that's seen this fly entering its territory. It's going to chase it. What is a pretty dramatic sequence. Now, the thing that really interests us about this is the fact that all of this is happening very, very fast. And we're looking at insects that are flying at 200 to 250 times a second. We have to film them at frame rates that are very high. The actual duration of the entire sequence that you saw is, the order of a few eye blinks, really. And this question has interested us for many, many years, is how are these insects able to do something so precise and so fast? And how do you, actually get about studying them? Now, the flies that I just showed you are great systems of study, but they're a little small. So we need something larger. And the insect that is sort of the workhorse of the lab is this. This is now a hawk moth. It's called the oleander hawk moth. And what you're seeing it doing is feeding on flowers at night. Okay, so you can actually see that its eye is dark adapted, and it's able to hover, put its proboscis very precisely into the flower. I think Tanvi might talk about this later in greater detail. But this whole process is actually very, very interesting because the flight is very stable. And clearly there's a feedback process at work which involves sensing the environment and how it's changing when the animal is hovering in 3 dimensions, and then responding to it so that you stay in place. And simple actions that we do, for instance, if I walk across this lecture hall, involves a lot of computation and very quick responses. I have a postural system that is constantly responding, making sure that I maintain my posture as I walk across. I'm looking at something, so my brain is responding to the visual stimuli. I have an inner ear system that makes sure that I remain in balance. If something goes wrong with it, I'll sort of I'll be giddy and I'll fall. So all of this is very important. Now, you take this question, speed it up, and then put it in 3 dimensions. That's really the question that we are looking at here. And this, of course, involves many what we call behavioral modules. This is more a conceptual thing, a behavioral module, and I'll explain what these are and how we go about studying them. But if you take this moth now, and you look at various things that it is doing when it is hovering, on the one hand, it's positioning its antennae. You'll see towards the middle of the talk why that is important. It has to stabilize its head. It's an interesting thing. The moths have eyes that are stuck to their head. So if they want to stabilize their gaze, they have to move their whole head. This is true for birds as well, and bats. So they have something called a head stabilization response, which ensures that their gaze is stabilized. Then the wings have to move together, and then the abdomen has to flex. And some of these are, mechanical, mechanically connected systems. Is something that Tanvi showed in her PhD thesis. So what we do is we typically go outside, look at insects, what they do, find something interesting, bring it to the lab, and then try and understand it at all levels, from mechanics to the nervous system. And a lot of what I'm going to talk about today has more to do with the nervous system than the mechanics. Now, we know a little bit about how insects balance during flight, and that's through work done on flies. The video that I've first showed you are flies. So flies are, they belong to a category of insects called Diptera, which means they have just one pair of wings, just two wings. The hind pair of wings have converted into these structures called halteres. And these halteres are vibrating, typically antiphase to the wings, and they act as Coriolis force sensors. So, what they're doing is maintaining their angular momentum, and when the insect turns, they try to maintain the plane of oscillation. They experience Coriolis forces, which then are detected by sensors at the base of so there are these sensors at the base of their halteres. These are mechanosensors, and they sense the strain on the shaft of the haltere, which they then communicate to the brain. And this happens extremely rapidly. The only other insect that we know that has a structure like this is called the Strepsiptera, which is a very small order of insects, where the front wing have turned into halteres. And there's exactly one paper which has actually looked at this and studied this in any level of sophistication, not too much actually. Every other insect, so insects are a very large group, as you know, there are more insects than all other animals put together. Diptera are a hyperdiverse group, but so are many other groups. And only Diptera have halteres, and then this small group of Strepsiptera. And I'll just quickly give you a sense of how this works. So, this is a normal fly. As it's flying, you can see that it's doing a lot of wonderful things with its wings as it's taking off. Now, if you go and clip the halteres off, then what you see is a fly that can fly, but it has absolutely no control on its flight. Okay? So it's flapping its wings, it thinks it's flying, but it's actually got no control on it. It's sort of like what would happen to us if we damaged our inner ear system. Now, I mentioned that the halteres are only part of this dipteran group. They're not part of any other group. And so there was a question in the field for a long time as to how other insects are balancing themselves in flight. We all know that the sense is required, whether halteres give it or something else. That sense is required. And so the question was, how do they balance flight? And so many years ago, through work done primarily in University of Washington, where I was a postdoc, we found out that the antennae are serving a similar role as halteres in hawkmoths and possibly many other insects. And specifically at the base of the antennae, there are these mechanosensors, they call the Johnston's organs, which are sensing the movement of this distal part of the antenna relative to the basal part. And they're keeping very fine track. And what I'm going to tell you about is this particular organ and wonderful things about it, but also the challenges of working with it. But we'll step back and just look at antenna as a whole. So this is the insect now, the head of the insect, the front part is taken out. You can see that there's a brain in there and an antennal nerve that goes into the antenna. So that's the base where the mechanosensors exist. And then the mechanosensors consists of two major components. One is called the Johnston's organs, as I've just mentioned, and the other is called the Bohm's bristles, which are bristle structures on the surface of the antenna. You can see them sort of here and there. And so many years ago, a graduate student, my first graduate student actually, Anand Krishnan, looked closely at this problem along with some of his colleagues. And what he showed was that when insects take off, they position their antennae. Okay, so you can see that here. So just before it takes off, that first behavioral model comes into play. The antenna is positioned. And we looked very closely at this problem, primarily to ask how does the insect know where its antenna is? Because everything is happening very fast. And to make a long story short, those are the Bohm's bristles right there. Look carefully somewhere here. And you'll see that as the antenna is positioned, somewhere there, you'll see those bristles brush against the joint right there, and that constitutes the signal to the brain that we then looked at, and that part of that stimulus goes into a part of the brain called the antennal motor and mechanosensory center, where it closely interacts with the motor neuron. Now, motor neurons are neurons that connect to the muscles. So what is happening is these bristles get brushed, they send the signal to this part of the brain where they synapse with the motor neurons, which then sends a corrective signal to the muscles. So if you take this antenna now and you move it, it'll very quickly come back to its original position. And this is something that we looked at very closely through a number of electrophysiological studies. So this is one setup by Anand Krishnan, Sunil Prabhakar, and others, where what we did was we sort of took a stepper motor and we stimulated these bristles. And while we did that, we recorded from the muscles. And we showed that the latency of the response, so from the time you stimulate to the time that the muscle responds, these are, this is the stimulus. This is a raster plot, meaning there's a lot of responses here, about 60 trials, and each of these are spikes. And you can see that they line up pretty nicely following the stimulus. And the difference between them is literally on the order of less than 10 milliseconds. It's extremely quick. So what's going on is that you stimulate these bristles and the muscles very quickly respond to it, and the antenna comes back in position. And the antenna needs to be in position because, as you'll see, the antenna is involved in flight control. That antennal position, as it turns out, has many other functions as well. And Taruni Kurana Roy, working with bees, put bees in a wind tunnel, and she asked the question, how do the antennae respond when you change the frontal airflow? And so what she does is she sort of digitizes the antennae and looks at how the angle changes. And what she saw was something interesting. So as you go from small airspeeds to larger airspeeds for this bee, the antenna comes closer and closer together. Okay, so the antennae come further and further up. And this is something that is a very characteristic behavior. You can do a number of trials and see the same behavior. Turns out this is also what moths do. So here's a video in which we change the airflow. And as we change the airflow on this tethered moth, you can see that the antennae come closer and closer together. This is work done by Dinesh Natation in collaboration with Nitesh Saxena. And there, again, you see those similar sigmoidal plots. And this seems to be mediated again by the Johnson's organ, the same sensors that I mentioned earlier. And if you restrict them, this behavior goes away. So for instance, here's where only one, the Johnson's organ on the left are restricted. And here, both Johnson's organs are restricted. And you can see that the antenna does nothing even when the airflow is going higher and higher. And so based on all these studies, we came up with a model. And in the model, the Johnson's organ receives information from airflow and other modalities. And then what it does is it sort of communicates with an inner loop. And that inner loop contains of the Bohm's bristles and the antennal muscles, which ensure that the position remains the same. And subject to airflow, that position can then change. So there's a very controlled change in that position, and the moth then knows exactly what their antennae are doing. So this is just a summary of everything I've just told you. I won't belabor it. Just one thing to add, which is that position is also a function of, I said, other modalities right there. This includes the visual modality, meaning if they see something, a front to back movement on the eye, that will also cause the antenna to change position. But interestingly, a front to back movement, which suggests that the moth is flying forward, causes the antenna to go back. Front to back airflow, which also suggests that the moth is going forward, causes the antenna to go forward. So there are these two balancing reactions going on, which keeps presumably the antenna in place. Harshada and Madhuri more recently have been looking very closely at the anatomy of these, and so largely speaking, what we see, so these are the Johnson's organs, which not been easy to fill, but we've been able to do it through a lot of conversations with people all over the world. The information from Bohm's bristles goes again in the same region that the Bohm's bristles were going. So the Johnson's organs are also sending the information to the antennal motor and mechanosensory center of the brain. There's a certain region of the brain. Now let's look at a little bit of the physics of what's going on. So when the insect is flying, the antennae are vibrating, okay? And they're vibrating primarily at wing beat frequency. They're probably like cantilevers. And so we wanted to know what the strain patterns were at the base of the antennae. It's almost, I don't know, I won't say impossible, but it's very difficult to measure any of this. So what we did was we modeled this using finite element analysis. And I think what you can see is that for every oscillation, the strain patterns move 2 times. And those are Coriolis strain patterns. And we know that the magnitude is high enough for the Johnson's organs to sense. So we wanted to then record from the Johnson's organs. So this is the setup. These were experiments done with Alexandre Dieudonné, whose picture you'll see in a little bit. So you have the moth in a tube. Its antennae comes out. It's inside a glass capillary. Then we move it with a servo motor. We know exactly what we're doing, but we'll question that in a little bit. And then there's we record from single axons of the Johnson's organs in the antennal nerve. And so what we then have is a spike record in response to a stimulus that we know. And so we can then correlate those stimuli. And you can see how precise all of this is. This is Alexandre Dieudonné. And you can see that if we give it a nice oscillatory stimulus, just jiggle the antenna back and forth, the spike rate increases in very regular, very correlated manner. And you can see that that's the... Now, if you give it a chirp stimulus, so if you want to know exactly to what frequency regime this is tuned, you can give it a chirp stimulus, and then you get a spike pattern. And what was interesting here was there was a region of the chirp stimulus, of the response to the chirp stimulus, where the fidelity of the response was extremely tight. So the gray is standard deviation, and you can see just how tightly correlated those responses are. And what that means, presumably, is that these two are, this particular unit is tuned to somewhere between 60 to 70 hertz, which is exactly twice the frequency of the wing beat, which is exactly where the Coriolis forces were, were transmitting information. When we do enough of these, we find that that tuned region actually is different for different units. So for many units, it's in this twice wing beat frequency regime. But if you look across Johnson's organs, then some of them are tuned there, some of them are tuned there, some of them are tuned there, some of them, and so on. And this is very beautiful because this then makes this Johnson's organ extremely versatile. It is able to encode some low frequency stimuli, some high frequency stimuli, some mid-level stimuli. So it is certainly encoding in all of these regions. And this makes it a really versatile organ. It can sense everything from gravity to airflow to to vibrations of the antennae, the Coriolis forces, and so on, which is why this then becomes a really interesting organ to study. It's also extremely sensitive. In fact, we never quite got to the point where our instruments were good enough. We could go as low as 0.05 degrees of oscillation, but below that, we didn't have the precision to determine, what the exact. And they were responding, or they just just about drop off there. But they're extremely sensitive and extremely fast. So the latency is on the order of 3 milliseconds. Now, when we study this, typically what you do is so the problem is this: you have a neuron, you want to characterize its frequency response, so you poke it with an electrode. And the moment you poke it with an electrode, you're going to kill it eventually. So now you have a limited amount of time in which to characterize this neuron. And you can't give it sinusoids going from 0 to whatever, 100 hertz, several trials each, etc. I mean, by that time, the neuron will be dead. So what you do instead is give it something called white noise, which is a broadband stimulus. More precisely, it's band-limited Gaussian noise. So, power is equally distributed in that spectrum. And so you give it some sort of a white noise. It's not exactly white noise, but this is one of the trials, and you can see. And so what you then do is for each spike, you ask what was the stimulus that just preceded it. And you do this over and over again until you get a large number of records. Then you ask what's the average stimulus that created or that generated the spike. So that's the average stimulus. And then you take a Fourier transform of that, and that gives you the frequency, the tuning response. And so it's using techniques like this that in the previous case, in the case of the experiments where we recorded from Johnson's organs, we showed that there were a large number of units tuned to, again, this sort of twice wing beat frequency regime. So the final step then was, of course, to ask, well, what happens if you actually stop the Johnson's organ from getting any stimulus? So this is a normal moth. This is now a behavioral experiment. So these are normal moth flying around. These are very old experiments, but I'm repeating them because this larger story is very important. And also the point I want to make is that nervous systems cannot be studied in isolation of behavior. You have to study the whole thing. Otherwise, you don't get the whole story. So this is a normal moth just doing what moths do, just wobbling around, flying. Now if we go in and we clip off the antenna, so this is the antenna. So the mechanosensors are here. So you can make a cut here without actually damaging any of this. And so that's what we did. But when you do that, the entire load is not anymore being sensed by the Johnson's organ. So you effectively reduced the stimulus to it. And that's what's been done here. We've got rid of the flagellum, and now the moth has exactly the same problems that you saw in the flies in that when we clipped off their hot ears. So they collide, they crash, they are not able to fly. And this is just a quantification of that. Now, you might say that, well, it's a pretty invasive experiment. The moth might be in pain or whatever it is. So you need a control experiment. And the control experiment is to glue the antenna back. Okay, and this should, it was a crazy thing to do, but it worked. So we glued it back, flight was restored. It's not perfect, but it was, pretty good. And so based on all this, there's no, so we are not looking at airflow here. It's just the vibrations. And that's mainly the stimulus, yeah. But of course, there's also some airflow related signals which we are not really looking into here. So there's a mechanics part of it. What constitutes a mechanical stimulus experienced by the antenna? It's the Coriolis stresses. Then there's the neurophysiology part where we know that we have neurons that are tuned to Coriolis frequencies, and then behavior. If the moths, the stimulus is removed, they cannot control flight. If you glue it back, then they can regain flight control. So this stuff is old. This is a long time ago. We've been thinking about this for a long, long time. One of the problems I have always had was that the broad approach that we used was this. It's what you would roughly call the black box approach. This is your neuron. You don't know what's inside it. And what you do is you give it inputs, you give it all kinds of inputs. That's a white noise analysis. And then you look at the output and you try and correlate the output with the input. In a little bit, I'll tell you that there are problems with this approach, one of which is our assumption as experimentalists that we are giving a precise white noise. And it turns out that that assumption is actually a fatal one. So there are other approaches, something one can call a gray box approach, in which you make some specific hypotheses and then go in with those hypotheses and test them. And that turns out to be a little more fruitful. So the caveats with the white noise analysis, one is, as I mentioned, the fact that, so this is a whole mechanical system. It's got linkages. The antenna is inside the capillary, and then you're moving the capillary, presumably what you think is white noise. But the whole mechanical arrangement filters that stimulus. So the stimulus that we actually deliver, this is what we would like to deliver, and what we actually deliver is something quite different. And we know this because what we did in a later version of the experiment was to attach at the base of the antenna a sensor that actually looked at what the antenna was doing in response to this movement. And it turned out that it wasn't doing exactly what we wanted it to do. I mean, it's not terrible, but there are problems. So it's essential to actually read out the stimulus to the best of one's ability. Again, this is not going to tell you exactly the stimulus that is being delivered, because the Johnson's organs are sort of sitting in there while the antenna is being moved, and we don't know what each of those units is experiencing. So we have just a very broad view of what might be going on. Yeah. So there are problems, but this is better than the previous version. So this is what we went with. This is Chinmay Mukunda, who just graduated. And what we did was try an early version of what one might call this gray box approach, which is to ask specific questions. Does it encode position? Does it encode velocity? And so then you design the stimuli such that those stimuli are simpler. They're not as complex as white noise, and so you can actually see that the stimulus that is delivered is the same as the stimulus that you intended to deliver. And so you can have a constant velocity and varying position stimulus if you want to test the hypothesis of whether it encodes position, or constant position and varying velocity if you want to look at the velocity hypothesis. And then you can do some variations of that, such as the stare stimulus, in which both the velocity is kept the same, but you vary the position. And this is just sort of a quick glimpse at the kind of data that Chinmay got. This is all published. I'm not going to go into too much detail with her data because that's a whole other talk. But I wanted to bring in this into the discussion, this idea of white noise analysis and the problems with it. And hopefully get from dynamical systems alternative approaches of dealing with this problem. Yeah, so I'll just summarize Chinmay's work, which is in this paper. The primary sensory neurons of Johnson's. Oh, I'm ready.
So
okay.
Just divide
I'll just finish this. I will just finish right here. The primary sensory neurons encode movement of flagellum and derivatives of movement stimulus. The Johnson's organs linearly encode angular position in the steady state, firing state. The problem is the neurons have a dynamical response, they adapt. And so all of those will confound the white noise approach. And so that's really, um, sort of something that causes problems. And so ideally one would like to look at the response and be able to predict the stimulus, but we are not able to do that because there's fundamental problems with the white noise analysis. So I'll stop there. There was a lot more, but just hmm, thank the lab. That's the lab on a particularly busy day when many people were there in summer and summer interns and so on, and my funding agencies. Thanks. Doug.
The antenna ultimately provide that's important for flight. So do wing beat patterns change in response to all the information that the antenna is providing?
Yeah, so that was the part I glossed over.
Okay.
But in a series of experiments, we've recorded from the descending neurons. And what we show is that even in the descending neurons, so information going from the head where the antennae are, from the AMMC there, down to the thoracic ganglia where the motor neurons are for the flight muscles, you can actually get recordings in the descending neurons which correlate with antennal movement. So that information is being sent down to the flight motor system. And so we now, I think that's some evidence that that information is available to the muscles that drive the wing motion. So as to what information that is, that's a very difficult question. But certainly the Coriolis band is visible in the descending neurons, so that there's also it's also transmitting in that regime, in the twice wing beat frequency regime.
Then twice the wing beat frequency, the fact that so many neurons are tuned to that twice the wing beat frequency, again, what's the why so many neurons? Is that sort of like a phobia there for that particular frequency?
Yeah, well, it's not used to thinking of phobia in mechanosensory sense. Yeah, but something like that. It may be, like I said, if you look at all the units or a large number of units, they vary in tuning. Right. But there are a large number that are tuned.
But which is why I was asking. I was comparing it to a phobia because
no, it makes sense. It's a nice thing to apply. I've just never thought in those terms. But maybe it could be that there's a whole set of Johnson's organs that are scolopedia that are just tuned to that particular response because it's needed for flight control.
And would that then, if it's mechanical, would that change with the size of the moth? Maybe in a different size moth, would you see a...
Yeah, that's a really good question, and that requires us to do everything that we've done here with other moths. And the heart is willing, but the clock is ticking, so to speak. But maybe Tanvi can take that forward.
Thank you. Very nice.
Yeah.
Two questions. One, I thought the tuning was more about fidelity rather than response amplitude because there were other frequencies at which responses were, right?
It's also about fidelity. So in fact, stuff I've left out is to look at jitter. And the jitter is really tight, on the order of 0.5 milliseconds or even less. So the fidelity is extraordinary. And that's something that, so the fact that the sensitivity is very high and fidelity is extraordinary is something that makes this, I mean, it's a really super organ in many ways. And if you think this is impressive, actually, mosquitoes are far more impressive than this. And their antennae are just, I don't know what to call them. They're hyper exaggerated, Johnston's organs. They can't even contain it. So there's like a donut shaped base. And mosquitoes, as you know, are able to fly in the dark, complete darkness. And so, that might be derived largely from feedback from the antennae, something that a graduate student in the lab is looking at more closely.
Yeah, the other question is, so when you said about the white noise analysis, so the error is coming from because the applied white noise is directly not coupling to the point where you want it to be white, or also
the error is
high
coming
non-linearity within the system, which I
both. So both of them. And so for white noise analysis to work, the stimulus really needs to be white. So first problem is that in almost no experimental system that I can think of, can you deliver that kind of a stimulus? Because it's invariably going to be filtered. So that's one problem. And so at least you need to know what it was that you delivered. And that's as far as we've gone, right? The second part is that the neurons themselves are dynamically changing their response. So if you give it a step stimulus, for the first part of the step, it will respond differently, but for the later part, it responds differently. Yeah? Exactly. So the basic assumptions fail. It's not a stationary system. So yeah, I mean, so we I think with some pain are ditching the white noise approach and trying to look at alternative approaches.
About the physics of light, these animals. So I guess in the downstroke, you transfer momentum to the air, but then how does the upstroke work? How does it compensate it? And how is that controlled by the animal?
Yeah, so that's a whole other talk on aerodynamics of insect flight. So actually when they hover, the wing goes sort of like this, where they're constantly pushing the air down, and then it flips, and then goes like this, where again they're pushing the air down. So they're actually generating forces on both strokes, upstroke and downstroke. So it's not upstroke, and it's more, like this. There's a few insects that do this, and in those cases, like hoverflies, for instance, will actually generate all their lift on the downstroke and none at all on their upstroke. So they just go at almost zero angles of attack when they go up.
So you control both strokes the same motor pathway.
Yeah. It's complicated. So at the base of the wing, there's a large number of muscles. In the case of flies that we've looked at, something like 18 pairs of muscles that are controlling. It's a concert going on with the nervous system telling them, you fire now, you fire now, you fire now. And so when the wing motions are being controlled, they're controlled at extremely rapid rates, but very precisely. And that's really, why this is such an exciting problem. Because one normally assumes that if you speed up something, you lose precision, but not in this case.
There are questions.